Okay, so the olfactory system is actually quite a remarkable system anatomically. There are bones in the nose, actually very thin bones. They're actually quite strikingly beautiful. I have a coyote skull here, and I'm going to see how close I can get so you can actually see inside the nasal cavity of a coyote, and you can see all those absolutely stunning. They're almost fractal. They kind of fill that space, and they're called turbinates or turbinals, and they're coated with olfactory neurons. And they are actually there's an epithelium, like a layer of cells over these bones upon and these include like those bipolar sort of olfactory odorant detecting neurons. And I have an image here actually on our site that's of it's a cross stitch by Steph Bowen of embroidology. I have a link to her work. Actually, you can Google it if you'd like to. She does some really remarkable, science art, and it shows you, for example, at the lower portion of this cross stitch, all of the various odorants, all the volatile chemicals that you're breathing in, to these nasal cavities, and they're wafting around those turbinates, and some of them are going to bind to specific odorant receptors, actually, and you can see they're indicated there. There's like these little projections out into the nasal cavities. Those are effectively, functionally, the dendrites, the receptive end, right? Then there's the cell body. There's one pole going to the cell body. And then the other pole, look at it. It actually goes up through the bone, the actual bone. It goes, it turns out there's a part of a bone at the base of your skull called the ethmoid bone, right above the nasal cavity, is a portion of that ethmoid bone that's called the cribriform plate. And the cribriform plate is sieve-like. It has lots and lots of little holes. It's like a colander. And there's lots and lots of these, projections from olfactory receptors that are localized in the nasal cavity, sending their information about what they detected in terms of the volatile odorants in the nasal cavity back directly into the central nervous system. So these series of axons, basically functionally axons from the bipolar odorant neurons that are found in the nasal cavity along those turbinates. All of those are forming what we call the first cranial nerve number 1, or the olfactory nerve. And it's kind of a weird nerve in the sense that you think of a nerve, being a single cable of things, but this is like lots and lots and lots of little cables that are going through all those various holes in the cribriform plate of the ethmoid bone at the base of the skull there. And so what I want you to see, it's kind of interesting, the odorant receptors, they're indicated here in different colors. It turns out in the human about 400 genes actually are devoted to coding for, different odorant receptor molecules, proteins that can bind, different types of chemicals that are volatile that you're breathing into your nose. 400 is a lot of genes, given that we have about 20,000 genes. So, what's going on here? And also it adds to, a lot of different potential labeled lines in the nose. So what happened, the way that this system is organized is that all these different subtypes of odorant receptor, they're different based upon which particular sets of those 400 genes are being expressed, and so which particular odorant molecules that you breathe in that you can actually detect. Well, they're arrayed all along that olfactory epithelium, along those turbinates, sort of distributed so that they can, have good discriminative capacity. There's a large surface area, or larger surface area, of which they're all splayed out upon. And then yet, as their axons, the axonal poles go through the cribriform plate into your central nervous system, there's an organization that happens. Like the ones here that are indicated, let's say in yellow, all the yellow ones, or all the blue ones, they're distributed across the nasal epithelium inside the nose, but their axons will coalesce, will converge in this structure that's found along the base of the frontal lobe called the olfactory bulb. There's two olfactory bulbs, one over the left, nostril, and one over the right, right nasal cavity, left nasal cavity, right nasal cavity. And think of all of these converging, axonal wires coming from these, specific, subtypes of olfactory receptor converging on a cell that's found inside the olfactory bulb called the mitral cell. And the mitral cells are sort of gathering up like all the blue inputs, all the yellow inputs, all the red inputs, all the odor receptors that express, odorant receptor genes 1, 2, and 3. And here's all those that 3, 4, and 5 or whatever. And they're all converging and they're forming this kind of basket-like assemblage of synapses in the olfactory bulb in the central nervous system on the bottom of the frontal lobe. And these basket-like assemblages of the inputs of the wires coming in from the specific kinds, of odorant receptor in the nose and converging on the mitral cell, that you find in the bulb. This is called a glomerulus, and the plural is glomeruli. So the sort of functional unit, the organization of, collecting all of the, subtype A odorant receptor input, even though it's distributed across the nasal cavity, they'll be all over, and their axons are going through different holes in your Cribriform plate. They're going to all converge. All the ones from, subtype A are going to go over here to this glomerulus, and then that mitral cell is going to send its axon back towards the rest of the central nervous system. It forms what's called the olfactory tract, and there's one on the left and one on the right. So the bulbs are, further right above your nasal cavity there. Really, the olfactory nerve, is super short. It's the shortest cranial nerve. It's just going through the bone straight into the brain. And then it's, forming these glomeruli structures with the mitral cells in the bulb. And then the axons of those mitral cells are leaving the bulbs, left and right, and working their way back into the brain. And they're working their way back along what we call the olfactory tracks. And actually, what's really interesting about olfaction and kind of unique about olfaction, there's a lot of stuff that's kind of unique about it, but is that while all the other senses we're going to describe, there's an obligatory synaptic connection, synaptic stop in that central set of, structures, nuclei called the thalamus. So here is a brain stem. I'm going to put it back together here. Here's a brain stem, right? And we're looking at, the front of the brain stem. It goes up like that. And this would be the top portion of the brain stem, which is called the midbrain. And here's the insula you can see on either side too. But within here are two big egg-shaped, clumps of gray matter. This is the thalamus on the left and the right. And most sensory systems like gustation we already discussed, right, has to make an obligatory stop in a nucleus here before being sent onto cortex to the, frontal operculum and the anterior insula. Well, olfaction has privileged direct access into medial portions of the middle portions of the brain, ancient portions of the brain, limbic portions of the brain, and again, it's, projecting back ipsilaterally, same side. This nostril is going to, if I close this nostril is going to this side, this nostril is going to this side. Very ancient chemical sense, very unique in a lot of ways, very unique sort of short cranial nerve, number one, olfactory nerve, and also, a lot of receptor proteins that are expressed 400 in the human, and, just very, very unique. No thalamic stop on the way to cortex.